فيما يتعلق ب Biochemistry of the endocrinology. Today we are going to talk about something very easy, very simple to understand, basically because it describes the synthesis or the metabolism of certain neurotransmitters or signaling molecules. And what we are going to talk about or focus today is on the catecholamines and thyroid hormone synthesis. In fact, we already described how we synthesize thyroid hormone or how it's going to be metabolized. And I think it's going to be easy issue today to talk about. The new thing is the catecholamines, which is describing how they are formed and the precursors, in fact, of these catecholamines. So basically, these molecules or signaling molecules of catecholamines are synthesized in their final active form, if you would, stored in granules and then secreted to the endocrine system or the bloodstream upon the signaling by certain stimulators. So it depends on many stimulators in order to release such kind of neurotransmitters or signaling molecules. All of the catecholamines molecules are synthesized from one precursor, which is an amino acid and known as tyrosine, or sometimes they are saying tyrosine. It depends. So tyrosine can be also generated from the metabolism of phenylalanine, if you remember this story. Just to know that phenylalanine is not a precursor for the catecholamines. It's the tyrosine, which is the precursor for the these catecholamines, while the tyrosine itself can be generated from the phenylalanine. So, and there is a big story about the metabolism and the deficiency in the metabolism of amino acids. For example, if the body cannot convert phenylalanine to tyrosine, there is a problem in the accumulation of the phenylalanine and development of what's called phenylketonuria disease, if you remember. So, and basically, it's not our story today. Today, we are going to talk about the metabolism of the catecholamines themselves. So, in order to know what we are going to talk about, catecholamines describes or includes dopamine or norepinephrine and epinephrine molecules, or DOPA sometimes. So these molecules are synthesized in different organs. For example, the dopamine is synthesized by certain cells in the brain. Norepinephrine, which is the methylated epinephrine, in fact, synthesized in the brain and adrenal medulla, specifically in the chromaffin cells, while epinephrine itself, which is unmethylated norepinephrine, in another word, I'm sorry for that. Let me close the phone, basically. Okay. And 80% of the catecholamine, it represents, I mean, the epinephrine represents the 80% of the catecholamines in the medulla of the adrenal gland, and it's not made in the extramedullary tissues, and specifically in the chromaffin cells. So this is the step of the conversion of the norepinephrine to epinephrine. And what I want you to know is that there is a difference between the norepinephrine and epinephrine by having a methyl group on the amino group of the norepinephrine. Okay, and this is, or this reaction is catalyzed by methyltransferase enzyme. Here, phenyl methyltransferase enzyme, which is responsible for the methylation process. And of course, you need carbon carrier or carbon donor molecule in order to generate this step. And we will discuss this in details in the few next slides. Now, we have four major reactions that are responsible for the metabolism of catecholamines or synthesis of catecholamines from tyrosine. And starts with hydroxylation, the first step, decarboxylation, and then we have another hydroxylation on the side chain. And finally, we have unmethyl or unmethylation of the norepinephrine in order to form the epinephrine molecule in the last step. So let's go to have a look to the whole pathway of the synthesis of the catecholamines. So starting from the tyrosine, we have to generate what's called dopa molecule, and it's known as 3,4-dihydroxyphenylalanine molecule, okay, because it's similar to the phenylalanine and tyrosine as well. And then generation of the dopamine. The next molecule is norepinephrine, and finally we will have epinephrine. So here we have hydroxylase, decarboxylase. It means we have hydroxylation, decarboxylation, another hydroxylation in another position, as well as finally we have methyltransferase by the enzyme that we described before. So step by step, this is what's going to happen at the beginning. So we'll start with the tyrosine, and conversion of tyrosine to dopa molecule or 3,4-dihydroxyphenylalanine molecule, because we have these hydroxyls on the previously tyrosine molecule. So it means that you are adding hydroxyl group. Okay, and this step is catalyzed by tyrosine hydroxylase enzyme, and there is a feedback inhibition by catecholamines for this enzyme. That's why this enzyme is representing a rate-limiting step of catecholamine metabolism or synthesis. And you notice here the presence of what's called tetrahydrobiopterin, and sometimes it's abbreviated as, let me write that, okay, BH4, okay, that will be converted to dihydrobiopterin, okay. So this means this is a tetrahydrobiopterin, and this molecule represents the dihydrobiopterin, and you lost the hydrogens in order to have hydroxylation process, and this reaction requires the presence of oxygen. هلا ليش بركز على هاي الشغلات؟ لأنه مطلوب منك تعرف every step in the metabolism of catecholamines. You have to remember the enzyme, the coenzymes that are involved in these reactions. So it's going to be a very easy mission for you. So the next step is the decarboxylation step, as we described earlier. So we will have here the first step of the hydroxylation, and the next step is the decarboxylation, which means that you are going to convert dopa to be dopamine. And as we can notice here, the dopa molecule has carboxylic group here. So the decarboxylation of the dopa by dopa decarboxylase enzyme is going to remove the carboxylic group, and you will have amino group alone without the carboxylic group. So this process of decarboxylation will form carbon dioxide molecule. And usually when we have such kind of reaction, I mean as a decarboxylation reaction, we have what's called PLP. PLP اختصار ل pyridoxal phosphate, which is a coenzyme for most of the decarboxylation enzymes, for most of the decarboxylases. But decarboxylases in general, they require pyridoxal phosphate as a carbon carrier in order to have complete decarboxylation of the precursors or the substrates. And you will get what's called dopamine and amine. It means that the dopa now has an amino group, which is this one. Okay. So this reaction almost is present in all tissues, as you can see here. And methyl dopa can be used as an effective treatment in certain kinds of hypertensions. So there's a kind of use of this methyl dopa molecule in the treatment of the hypertension in certain cases. Now, the next step is another dehydroxylation in another position. And let me here move the, okay. But now it's beta hydroxylation. So the position of the hydroxylation is different now from the previous one. This is the first hydroxylation, and this is the second hydroxylation site. And the hydroxylation, of course, requires the presence of hydroxylase enzyme, and the precursor or the substrate is dopamine. So assumably we have dopamine hydroxylase enzyme, and this reaction will produce the norepinephrine molecule. As coenzyme molecule, ascorbate or ascorbic acid, is required for the catalysis of this reaction, as well as the presence of the copper in order to to hydroxylize the dopamine in order to produce the epinephrine molecule. Okay, so you have to remember and recall the rule of coenzymes in each of these reactions. So the next step, which is the final one, is conversion of norepinephrine to epinephrine. And as you can notice here, the difference between the norepinephrine and epinephrine is just the methyl group here, which will be added by phenylethanol-amine and methyl transferase enzyme, the enzyme that we mentioned before, اللي كان اختصاره PNMT. And in order to proceed with such reaction, you need carbon carrier, carbon donor. And the carbon donor in this reaction is known as SAM, or S-adenosyl methionine molecule. It's another coenzyme that is required in order to donate the carbon or the methyl group for such kind of reaction. And the methyl transferase is responsible for transferring the methyl group from the SAM, or S-adenosyl methionine, to form S-adenosyl homocysteine. So you are converting the methionine to homocysteine by demethylation or transfer of methyl group from this molecule to the norepinephrine in order to generate the epinephrine molecule. And finally, we have this complete portrait of the catecholamine metabolism from tyrosine, dopamine, norepinephrine, and epinephrine. So every single step in such or in this reaction is required to be memorized, and you're going to be asked about all of these steps. Just to have an idea about the role of the catecholamines and the replacement of the missing of these molecules, Parkinson's disease has been as a neurodegenerative disorder, and it's related to the movement phenotype. If there is an insufficient production of dopamine in certain types of cells, and it can be caused by idiopathic loss, which means that it's auto disease, we can replace the dopamine that already synthesized in the nervous system by administration of L-DOPA or levodopa molecules. And it's one of the most commonly used treatments in the treatment of the Parkinson's disease, as an example. Okay. The next type of hormone that we are going to talk about today, how it's synthesized. We already had an idea and discussed some steps of the metabolism of the thyroid hormones. And the most common forms that we are going to talk about, triiodothyronine and tetraiodothyronine. And we can generate actually the T3 from T4 by removing one of the iodines from the T4, which is the thyroxine, in order to produce the active form, اللي هو T3 form. And this process can be performed in the thyroid gland itself or in the periphery, which means that the target cells also have a potential to make the iodination of the thyroxine to generate the active form. So it applies for يعني هذا النوع من الهرمونز بنطبق عليه الحالتين اللي هو synthesis of the active form and released to the blood system or to the endocrine system, or it can be activated at the site of the action. So the synthesis of the thyronines in general, triiodo or tetraiodo, T3 or T4, already described before as a large precursor molecule known as thyroglobulin. So basically we have to have this peptide, which is the thyroglobulin, which is found in the colloid of the luminal side of the thyroid gland. And the cleavage of this molecule, cleavage of the triiodo or tetraiodothyronines from the thyroglobulin will generate the formation of T3 and T4, which are the active hormones, and the T3 is the high potential hormone. So this diagram actually has been discussed before, but today we are going to talk about general cyclization of the thyroglobulin, how it's going to be synthesized, as well as the disease that is related to the deficiency of the process of the metabolism of the thyroid hormones. So basically, the disease, the most common disease that is associated with the thyroid hormone synthesis or metabolism is the goiter disease. And we can see here the steps that are involved in the goiter disease if they are deficient. So basically, what we have to have in order to synthesize the thyroid hormones is to have the thyroglobulin peptide, which is already formed by thyroid cells. And it will be secreted by exocytosis to the follicular space of the colloid of the thyroid gland. And there will be a process of iodination of the thyroglobulin on the side chain of the tyrosine. So basically, these spikes represent the tyrosine that is found in the thyroglobulin peptide. And we need iodination process, which means adding of the iodine molecule. And before adding the iodine molecule, it should be oxidized from being ionic form in order to be iodine molecule as a molecule. Okay, so this process is activated by peroxidase enzyme, which is an oxidation process, and it is also it requires the presence of hydrogen peroxide in order to proceed with the conversion of the ion atoms or ions to iodine molecules. So later on, after iodination, we will have either mono, iodothyronine, or di, and then we have to generate either the tri or tetra form of the iodine or thyroid hormones. This process is generated by what's called coupling. Coupling means that you are going to transfer one of the tyrosine to another one in order, like for example, mono to di in order to have tri form, or di to di in order to have the tetra form. Okay, and you will have also di or mono, which are not representing the active forms of the hormone or thyroid hormones. So any deficiency in the process of the iodination, or for example, insufficient iodine production, will cause the phenotype of goiter disease, as well as the coupling. If we have a problem in the coupling of the tyrosines, which means transferring of one tyrosine to another one in order to generate the active forms of the thyroid hormones, also you will develop goiter disease. Now you synthesize the let's say coupled and iodinated thyroid hormones on the thyroglobulin. So the next step is to go to the bloodstream. So moving of the thyroglobulin precursor to generate thyroid hormones. حبيت احكي هيك لانه احنا بننقل thyroglobulin to the bloodstream in the next slide. You are transferring the hormones. So basically you have to engulf actually by phagocytosis process, or pinocytosis if you would, because it's small protein, to the thyroid cells back, and then we have lysosomal digestion, like phagocytosis process, in order to release the thyroid hormones in free forms, and then will be transported to the extracellular space. And after that, we will have thyroid transporter proteins, because water insoluble molecules, so that's why it needs certain transporters to be transported in the circulatory system. So the rest will be the degraded product of the peptide. هلا نذكر مرة ثانية إن إحنا لما نعمل lysosomal degradation you are degrading the protein itself اللي هو البيبتايد of thyroglobulin. So the rest of the molecules will be transported as either amino acids or the degraded products like the tyrosine, in order to be reused again in the generation of future thyroglobulin, as well as the iodine, which will be released as ion by deiodinase enzyme. And if you have also deficiency in this process, the goiter disease is going to be developed. Another cause of the goiter is the deficiency in the transportation of iodine by this kind of transporter. If you remember, we talked about the antiport transportation mechanism of iodine with the sodium-potassium pump. So if you have a deficiency in this transportation mechanism, the goiter disease can be developed. So the goiter is associated with either of these steps that we described before that are related to the metabolism of the thyroid hormones. So thyroglobulin itself is a large or considered as a large prohormone, if you would, for certain extent. فهو عبارة عن precursor للهرمون itself. Okay, and it's a large iodinated form, glycosylated protein as well. It means that it has certain glycoproteins, and these glycoproteins playing a role in the identity of the protein as well as sorting of the protein toward the site of the metabolism. And due to that, the carbohydrate represent from 8 to 10% of the glycosylated thyroglobulin molecule. The iodine representing up to 1% of the molecule. And you have to remember it consists of 2 subunits as a protein. And it is rich with tyrosine. We have 115 tyrosine residues in this molecule. And the ratio of the T4 to T3 on this molecule is like 7 to 1 on the thyroglobulin molecule itself. So these are the general characteristics of the thyroglobulin molecule. Now, talking about iodine or iodide metabolism, the thyroid as an organ is able to concentrate iodine ions against a strong electrochemical gradient because we have active transportation mechanism in order to transport the iodine. So that's why if we have high concentration, the transportation of iodine ions will be against concentration gradient due to the presence of the ATP pump. So thyroid serum ratio of the thyroid hormones or the iodine, concentration is 25 to 1. This means thyroid to serum ratio, okay, of iodine. Okay, if we have normal diet, of course, will be 25 to 1, which means that the iodine is concentrated in the thyroid gland, and this process is stimulated by the TSH hormone. Now, the thyroperoxidase enzyme, which is a tetrameric protein, which is responsible for the oxidation of the iodine to form iodide molecule, from ion to a molecule of iodine, is a tetrameric protein. And of course, it requires the presence of hydrogen peroxide, which is generated by 2 mechanisms. One of them is the hexose monophosphate pathway, and you have to remember that. And the other one is the malic enzyme, which converts malate to pyruvate molecule and vice versa. It's called malic enzyme. Okay. So we have source of hydrogen peroxide, so there is no worry about the insufficiency of the hydrogen peroxide. So the enzyme requires such kind of oxidizing agent in the process of the formation of the iodide molecule. Now, coupling means that you are going to form either T4 or T3 by either coupling 2 diiodine molecules or thiiodo-thyroglobulin molecules or monoiodo with diiodo in order to generate T3 molecule. هلا داي معناها انه في عندك 2 iodine فلما يكونوا اثنين مع بعض يكونوا 4 iodine, and here we have 1 and 3, 2, you will generate 3 iodine molecules to generate T3 molecule. Now, the deiodinase enzyme removes the iodine ions from the inactive mono and dithyronine molecules, which is the last step here. This is the deiodination reaction by deiodinase in order to recycle the iodine as well as the tyrosine. And we have as I mentioned before, a peripheral deiodinase activity in the target tissues in order to convert T4 to T3 as an active form in the target tissue. So until here, we discussed two simple issues, two simple metabolic pathways اللي هم الكاتيكولامينز بالإضافة للثايرويد هرمونز. And now we are going to move to something we already mentioned اللي هو activation of or formation actually of certain hormones by just cleavage of these hormones from a previous inactive or pre-pro form. البروتينات اللي بتنصنع بشكل عام حتى نحولها للactive form. Usually they are synthesized as pre-pro molecule, and then you will generate pro molecule from that, and then you will generate the active molecule. So there is a kind of a cleavage in order to generate the active molecule. And it's worth to talk about insulin as a hormone that is generated by the cleavage process. So basically the insulin is synthesized in this form as a polypeptide, and eventually there is a process of a cleavage at certain specific sites like this one here and the other ones here, okay? And the red arrow means that there is trypsin-like enzyme, which is responsible for the cleavage of this bond, the peptide bond, here and here. And we have also carboxypeptidase-like enzymes, which are responsible for the cleavage of these amino acids at these sites. So at the end of the day, you will generate this molecule, which consists of two polypeptide chains. And insulin is described as dipeptide or dipolypeptide or two-chain molecule, اللي هو chain A and chain B. Okay, chain A أصغر من chain B basically. هلا chain B is generated from the N-terminal pre-pro insulin or pre-pro hormone, while the A chain is generated from the carboxyl end. And this cleavage is going to be performed after the generation of or after the formation of the SS bond, which can be noticed here either in the same chain, that's why it's called intra-chain SS bond, okay, and the other one or other two types or two bonds, SS bonds I mean, that are formed between chains themselves. So this one is in the same chain and the others are between the chains. That's why these types of SS bonds are called interchain bridges or interchain disulfide bond. Okay. هلا لهم ارقام ولهم positions. انا مش كثير مهتم في هذا الموضوع انه is it for example A6 to A11 اللي هو موجودين في الchain A، فهذا يعتبر عباره عن intrachain SS bond, while the other two chains or other two sulfide bonds A to B at position number 7, and here from A20 to B90 or 19, sorry, in order to form the active insulin hormone. So this hormone is generated by cleavage as well as SS bond formation. Okay, the rest of the peptide will be called C-peptide, which is also secreted with the insulin, and it has for sure another function with the insulin, and we use it as a marker for the efficiency of the production of insulin from the pancreas. There's a kind of assessment of the efficiency of the hormone secretion from the pancreatic cells. So this is how we generate, for example, active hormone from pre-pro hormone. And insulin is the most common example that describes such a process. Another hormone can be generated by the cleavage of the pre-pro form, which is the parathyroid hormone, which is secreted as 84 amino acid peptide. But before being active, it has to be cleaved in order to have the active form. So the immediate precursor of PTH or any other kind of the inactive form of proteins is the PTH, but in fact it has a pre-pro pH, which is 115 amino acids residues or amino acids, the same thing. So the cleavage of the certain amino acids from the N-terminal end is going to generate pro PTH. So we have pro PTH. The cleavage of certain amino acids will generate pro PTH, and finally we will have the PTH as an active hormone. So the PTH1, for example, which contains 34 amino acids, I mean from 1 to 34, is fully active molecule, and they are saying, or it's known that the amino acids from, or the residues between 25 and 34 position are responsible for the binding function with the receptor. So small portion of that peptide is responsible for the binding, while the rest is just for the conformation of the hormone itself. Okay, so this is one of, actually it's another example of the cleavage of the pre-proactive inactive hormone in order to generate the active hormone. And this example here is describing the bovine pre-prothyroid parathyroid hormone, and this is the active parathyroid hormone or PTH hormone that is going to be generated after the cleavage at many sites in order to have the final fragment which is representing the active form. هلا هذا الجزء اللي هون اللي هو من 24 ل 34 is responsible for the binding of the receptor, while the rest is responsible for the conformation of the hormone itself. So the other part of the hormone is just for or sorting or any other functions can be related to something else related to signal transduction, but the PTH as a hormone or the active site is just located between these residues. The biosynthesis of the PTH and the secretion of the PTH is very well known to be regulated by the calcium ion concentration. And as you may remember from the physiology lectures, the reduction in the calcium ions can result in the increase in the PTH hormone by induction of the mRNA synthesis. So it means that there is a gene regulation process by the calcium that will activate the gene that is responsible for the synthesis, I mean transcription translation, or gene expression احنا بنسميه, of the PTH as a peptide, and the PTH later on will be secreted due to such kind of activation. Now, these are some of the, let's say, biochemical reactions that are responsible for the activation of the parathyroid. So there are a number of what's called proteolytic enzymes, like cathepsins B and D, and they are identified as parathyroid hormone enzymes, or parathyroid gland enzymes. So they are very predominant in the parathyroid tissues in order to have the cleavage. So cathepsin B is responsible for the cleavage of the PTH اللي هو pre-pro form to two fragments, while the rest of the molecule is going to be just kept as is, and the other molecule, which is the active form, is going to be cleaved in order to generate the active form by further cleavage. And most of the proteolysis of the PTH hormone occurs within the gland. So the hormone is almost processed in the target cell, I mean the generating cell, and then it will have further processing outside of the gland cells. So some studies suggest that the PTH, once secreted, it can be cleaved or degraded in the site of the action of the hormone itself. Angiotensin 2 is another example of the hormone that is synthesized as a full precursor molecule or pro hormone molecule. And the renin-angiotensin system, which is involved in the regulation of blood pressure, is very well known to be associated with the availability of the angiotensin 2. So the angiotensin 2 is octapeptide molecule which is generated from angiotensinogen. So we call it angiotensinogen rather than be called proangiotensin as a precursor. So you have to be careful when we call such molecules. The angiotensinogen, which is a large alpha-2 globulin that is synthesized in the liver, is substrate for the renin, which is the enzyme that is produced in the juxtaglomerular cells in the renal system in order to activate the angiotensin. And this is the axis of the renin-angiotensin activation or generation. So first of all, you have to start with the angiotensinogen, and the renin is responsible for the cleavage in order to generate angiotensin 1, and angiotensin 1 converting enzyme is going to form angiotensin 2, and followed by aminopeptidase in order to generate angiotensin 3. And finally, it will be degraded by angiotensinogenase enzyme or enzymes in order to get rid of the final product, اللي هو عملية degradation and removing of the hormone itself. Now, ACE, which is a hot spot or a hot story nowadays because it's related to the COVID-19 pandemic disease, because it's one of the the receptor of the ACE is responsible for the binding of the or recognition of the SARS virus, actually, or SARS-2. The enzyme itself, angiotensin converting enzyme or ACE, is a glycoprotein that is found in the lung or endothelial cells, in the plasma itself, which removes the carboxyl terminal amino acids from the decapeptides of angiotensin 1 to form what's called angiotensin 2 as well. Okay, and there are some kind of drugs that are targeting ACE mechanism, which are called ACE inhibitors, in order to treat some disorders. Now, we have what's called, as another example of the peptide hormone, we have what's called POMC, which is the abbreviation of Opiomelanocortin, as a peptide hormone. It's a family which consists of these types of hormones: adrenocorticotropic hormones, LPH hormone, MSH hormone. So these hormones belong to the same family, and they can, in fact, work as neurotransmitters or neuromodulators, if you would, like endorphins. And again, they are synthesized from pre-pro molecules that will be activated by cleavage of certain amino acids in order to have the active one. So just as an important issue to have an idea about, mutations of the MSH receptor are associated with early onset of obesity, as an example of the role of the MSH and its receptor in the obesity. And this observation has been noticed due to the redirection or actually redirected the attention to the POMC peptide hormones in order to target the treatment or to target the obesity as a disease by having such kind of alternative treatment. So this is how we generate the active forms from the POMC precursor molecule in order to have different types of hormones from one precursor molecule. So it's kind of saving the energy of the cell, and it will be more efficient to generate much more than one hormone or neurotransmitter molecule or effector molecule from one precursor in order to regulate the process or the whole process by just expression of one peptide rather than expression of too many genes. This is the most important thing that we have to talk about today. جزء منها معاد، جزء منها في نوع من الأشياء الجديدة اللي متعلقة بالرياكشنز. هلا في حد رفع إيده اللي هو رنيم.
دكتور، بدي أسأل سؤالين بخصوص المحاضرة هاي والمحاضرة السابقة.
تمام.
حكيت إنه في علاقة بالانجيوتنسين كونفرتنج انزيم والكوفيد، إيش العلاقة؟
الريسبتور.
إنه بيعمل له activation مثلا المريض بصير عنده hypertension.
هو جزء منه الاكتيفيشن because this enzyme الانزيم اللي هو الايس enzyme is can cleave the glycoprotein of the COVID in order to activate the infection.
ما
There is a
في
whole there is a complete mechanism describing this process.
يعني activation of this receptor بيساعد في الانفكشن أو
Activation of I mean the enzyme itself is going to activate the infection by COVID-19.
تمام. دكتور سؤال بخصوص المحاضرة الماضية. مذكور جملة إنه هو مكتوب إنه الكورتيزول synthesis requires 3 hydroxylases located in the fasciculata and reticularis zones of the adrenal cortex. هلا اللي بعرفه أنا إنه الكورتيزول synthesis بصير في الفاسيكولاتا ما بصير في الريتيكولاريس، فإيش المقصود هون يعني؟
الانزيم اللي responsible is localized also in two sides. Okay. هذا الانزيم موجود في المكانين هذول.
يعني
the final form of the cortisol, as you said, yeah, you are right. ولكن the process takes place in different places.
يعني بتصير في الريتيكولاريس وبتطلع تكمل البروسيس في في الفاسيكولاتا، هيك المقصود يعني.
تمام. طيب.
دكتور يعطيك العافية.
أنا كنت بدي أبلش بالمحاضرة اللي بعديها، هي رغم إنه محاضرة سهلة لكن I will leave it maybe we'll talk about it tomorrow. إحنا بكرة عندنا محاضرة اللي هو open ولا لا مش بكرة، هي flexible session. خلينا نشوف التايم تيبل. أنا مسجل عندي إنه 30 معناها مش بكرة، في عندنا محاضرة وعندنا 2 12 محاضرة ثانية. Okay, so next time will be 30th of November. Okay, let me see. راح تكون يوم الاثنين إن شاء الله الساعة 9، وفي عندنا محاضرة يوم الأربعاء 2/12. Okay, الساعة 10. Let me check. عندنا flexible lecture I think.